Welcome back to the course Fundamentals of General Immunology, and we are currently in module 2 and lecture number 4. The title of this lecture is Coordination between Cellular, Molecular and Tissue Level Immunity. So, we will continue with our last discussion to this chapter as well. So, we have understood this point of gene regulation when we discussed in the last class about central dogma, where the DNA of a cell can do a transcription to produce a messenger RNA, and this messenger RNA can do a protein synthesis by binding to the ribosome, and then this protein is actually secreted out in the form of a small messenger molecule. And in the last class we defined the term cytokines. And these cytokines can go and essentially bind to the receptor of the neighboring cell to do the effect. Now, essentially in immune system, when we discuss about the immune system or immune cells, particularly regulation of lymphocyte. So, in lymphocyte, we have two lymphocyte cells. One is called B lymphocyte and the other is called T lymphocyte. These two lymphocytes are highly regulated cell population, because these can produce, in later point we will understood, this will be responsible for antibody mediated immune response, mediated immunity, and this will be responsible for cell mediated immunity. And therefore, regulating these two cell types is critically important, and these are governed by a highly regulated gene expression inside the cellular system. So, if you see the genes which can code for the cytokine production are regulated by different transcription factors which are activated in a cell which are sensitized with the antigen, and this can regulate the production of a protein, essentially will do the cascade phenomena to ultimately do a effector function in the cell type. So, this is a general scheme of an immune cell regulation, exactly what we discussed in the last class concerning our understanding of central dogma in the tissue cellular level. All right. So, then again, this is just to recapitulate our details of the concept that once there is a ligand, and these ligands could be anything secreted out from the cellular system or coming from outside. This could be a chemical moiety, this could be any messenger molecules, and they can go and bind to the receptor of the cell. Now, receptor, as we discussed in the last class also, that these are specific protein which are expressed on the cellular surface. These receptors can give a docking to these ligands, and when there is a docking of the ligand with the receptor, it can do a downstream cascade, and because of this chemical messengers are produced in the cytoplasm, which in turn either do a response or can directly regulate the gene expression of a cell. So, this is one way the chemical messenger can do or trigger the reaction. The second way is when you have an exogenous stimulus such as pathogen, which can be bind or engulfed by a antigen presenting cell, and this could be a macrophages, this could be a neutrophil or any other cell type which are capable of engulfing the pathogen. So, once there is an engulfing of the pathogen by antigen presenting cell, this can give some signals or some kind of a stimuli to the neighboring cell population, so as to sensitize and give the response which can essentially do a kind of an infection could be aborted by using that response. So, this all response which are coming from a cell which received the signal from an antigen presenting cell is again a very synchronized process in a cellular system. So, we could also say that cell signaling leads to the production of signal that serve as inputs for the other cell populations. So, essentially these are two general schemes which we have to understand, that the regulation of the cellular system, the coordination of the cellular system is governed by either gene expression or by the production of different kind of messenger ligands which can bind to the receptor of the cells to give the effect, either to the same way which we call autocrine effect. It may give to the neighboring cell, which is we call paracrine effect, or which can go to the distant cell population to give an endocrine effect. So these are the things which we need to consider, and let's try to move on and see some more details about the cellular phenomena in the immune system. So again, getting into details of adhesion molecule, which we discussed again in the last class, that there are molecules which we call selectins, and these selectins are actually the specific kind of proteins which are present on the cell surface, and these are carbohydrate binding adhesion molecules. That means they can bind to the carbohydrate specific enriched areas in the cell and the tissue level. So there are classification of selectins, and for time being we are just trying to understand there are three types of selectins. P selectins, as name itself indicates, P stands for platelet. It was first discovered in platelets, but its expression is largely seen in endothelial lining of the blood vessels. Then we have E selectins, which are actually, name itself indicates, it's in the endothelium, while L selectin is actually expressed in leukocyte, which is white blood cells. Now, looking into the primary ligands, the ligands for these P-type selectins is actually P-selectin glycoprotein ligand 1. So, PSGL1, that's a ligand for P-selectin, which we will see in the details when we encounter that how these selectins are modulating the cellular trafficking or say migration of the leukocyte. Then we have E-selectin, that's either having a ligand PSGL1 or ESL1. ESL stands for E-selectin ligand 1, and CD44. Now, just to give a brief understanding about what is CD, CD stands for cluster of differentiation. Now, every cell in a human body is again identified by a unique kind of protein which are expressed on the surface of a cell. For example, if it's a liver cell will express a particular protein that is a hallmark of a liver cell. It may be present in other cell type, but its expression is exclusively noticed in liver cell. Then this molecule or this protein could be an identifying feature for a liver cell population, and therefore these proteins which are present on a specific cell type could be a signature molecule to identify that cell type, and therefore these proteins in immune system particularly, they are called a cluster of differentiation, CD, and they are named by their numbers 1, 2, 3, 4. So therefore you can see here the ligand for E-selectin is CD44. That means this is cluster of differentiation 44. That means any cell which is expressing CD44 can bind to E-selectin, or E-selectin could be a say target for that particular CD44 specific protein. And finally, the L-selectin. L-selectin has name itself saying it's a leukocyte specific selectin. So the primary ligand is CD34, and then we have a glycan bearing cell adhesion molecule 1, and also mucosal addressing cell adhesion molecule. These are all typical proteins which are expressed and involved in the cellular adhesion in the migration process of a cell. We can also look into that these P, E and L selectins are categorically said CD62P, CD62E and CD62L. P, E and L is actually demarcating their types, and then these are actually cluster differentiation. So one can easily say that CD62 is a specific marker of a selectin, and then we can look into their ligands and location categorically to understand how these adhesion molecules are delineated in the immunological system. Coming to the next part of adhesion molecules, after selectin we also discussed about integrins, and these integrins are actually heterodimeric cell surface protein. What do we mean by heterodimeric is suppose there is a cell surface and these proteins are actually having two polypeptide chain. One polypeptide chain could be A and the other polypeptide can be B. So these are actually a cluster of these two polypeptide chain. This could call as heterodimeric because A and B are different. If there is A and A, we call it homodimeric. If it is B and B, we also call it homodimeric, because both the chains are similar in nature. But here it is not similar, and therefore these are called heterodimeric surface proteins, because these are expressed in surface. Remember again one more thing, that these all proteins are heavily glycosylated. That means after production into the cytoplasm, these proteins goes to endoplasmic reticulum or Golgi apparatus to do post-translation modification, and there it gives, the protein gets a conformational change by the addition of different kind of sugar moieties, which we call glycosylation. This could be N-type glycosylation or O-type glycosylation based on the pattern of amino acid sequence present in the protein. So if it is N-type glycosylation, mostly takes place in endoplasmic reticulum. So N-type is mostly in endoplasmic reticulum, and O-type is mostly in Golgi apparatus. So this is not a universal way to depict, but mostly these are the two glycosylation processes happening into different organelle of the cell. So there are three types of integrin which we have listed here. One is called leukocyte functional associated antigen 1, LFA1. The second is, since it is an heterodimeric, so one chain is alpha, the other is beta, alpha 4, beta 7, that's a second type of integrin. And the third one is very late antigen 4. This is just a name of a protein. So these are all integrin which are again part of a adhesion system. If you closely look into their location, they are expressed either in leukocyte or the very specifically T cell and monocyte. So these are just a location wise details of LFA. So LFA1 is in leukocyte, alpha 4 beta 7 integrin is in monocyte and T cell, while VLA4 is in monocyte and T cell. What are their ligands? And the ligands is ICAM1, VCAM1. So ICAM1 here is actually intracellular adhesion molecule 1, while VCAM is vesicular cell adhesion molecule. That means it could be a protein which is present in vessels, which are vascular specific thing, and intracellular is just cell to cell communication. So one can easily see that these are specific adhesion molecule such as selectin and integrin, which takes part in the cellular adhesion, migration, rolling and all, which we have discussed in the last class as a diapedesis phenomena. So these are the brief details about selectins and integrin as an adhesion molecules. All right. Looking into how these integrins are activated, that is also very critical, and we need to go in little more detail and the molecular details of integrin in order to understand how these molecules are activated upon releasing or binding to a ligand. For example, here we see the inactive integrin. So inactive integrin is actually in the bend form. So we see that alpha and beta, these are two heterodimeric chains of the protein. So one is alpha and the other is beta, and there are two proteins which are very critical in activating this integrin molecule. One is called taline and the other is called kindlin. So this taline and kindlin are the two very integral protein that activates this integrin. So once this get bind to the region of integrin, they get straightened up, and that they call it an active extended conformation. So the protein is actually conformational in nature. So once there is a binding of taline and kindlin, then the protein gets extended. An extended protein can give a docking to the ligand. That means before that the integrins are inaccessible to the ligand. Once there is a binding, then a ligand could easily dock to the area where there is a pocket, binding pockets, and therefore this ligand can dock into the region of alpha and beta, and therefore this whole bunch of binding and the taline and kindlin expression or the binding to the cytoplasmic tail of integrin can do a cascade where there could be either possibility of actin cytoskeleton assembly. So we have multiple cytoskeleton proteins such as actin, myosin or cytokeratin, and these can be activated by these processes of integrin activation. And we could also see that this could lead to downstream signaling cascade as well. So once there is a binding of ligand to the molecule, then we could see that there is a clustering happening. So the other molecule which are close by can do a clustering, and then we can see extracellular modeling could also happen in the cellular system. So what it suggests that the activation pathway of the molecules in the immune system are critically regulated by lot of channeling molecules. Here we have seen that there is a molecule, the protein, which is heterodimeric in nature, and this is not in the active form, it's in a bent form. Once it is binding to a XYZ protein, it is getting extended. Then it is binding to a ligand, then it is getting clustered. And finally, it gets an activation of say some processes such as actin assembly, or for example any downstream signaling of the molecules. So this cascade is at most important to regulate the pathways, regulate the cells, and these are all critically regulated. When we are discussing this integrin adhesion molecule and activation of integrin, we need to understand that the cellular expression of these integrins are critically regulated, and any deviation. So for example, in the last class we discussed that there is a phenomena called diapedesis, and in diapedesis this integrin and selectin combination can roll the lymphocyte into the blood vessel, which can lead to the extravasation of the leukocyte towards the tissue area or lymph node. Now, if there is a less expression of selectin or integrin, or there could be modulation in the expression of these two adhesion molecule, we could see deviation in the migration, and therefore the immune response of the human being could be compromised. So the regulation of these adhesion molecules are also critically important. All right, we will move on, and we will move to the various important term called homing. Homing literally stands for going back to the home. So let's understand this phenomena, and then I have given here an example so we could understand that example once we understand the phenomena. So we have seen in the earlier classes that the T cell is actually produced in organ, primary lymphoid organ called thymus, and this thymus can produce, and then the T cell could come into the blood circulation, and from there it goes to the secondary lymphoid organ, which is a spleen, lymph node, or other areas of lymphatics. So a spleen and lymph node can get these naive T cell actually. So we can call it naive T cell. That means these cells are not activated in a way that, so these cells are not activated in a way that can cause its activation. So these are called naive cell, and once they are coming into blood circulation, they go and resident themselves into spleen and lymph node. Now if they encounter any kind of a pathogen, then they are pathogen activated antigen presenting cell, they will become activated. If they are not activated, they will come back into the blood circulation, right? And then this process is continuously happening into the system. This is actually called lymphocytic recirculation or lymphocytic remigration. Now, because of this phenomena, you constantly see a flux of naive T cell population in the bloodstream from the site of its production, which is the thymus, to the secondary lymphoid organ, which is spleen or lymph node. Now suppose if there is a possibility that there is an antigen presenting cell coming into the picture, and if there is an antigen presenting cell which is expressing this antigen on the surface, these naive T cell which are circulating in the blood circulation can identify this antigen loaded antigen presenting cell and could get activated. Now this particular cell which sensitized with the antigen presenting cell containing antigen will now called as activated T cell. Now it is not called as a naive T cell, it is called activated T cell. Now this activated T cell is required to go and do its effect. Now suppose if this activated T cell can go everywhere, then you see a rampant immune response. But if there is a requirement of this T cell, activated T cell, for example in liver or for example in kidney, then we can see that these specific activated T cell can go to a particular site only and not to a uniform circulation. So they can express certain kind of a receptor, and these receptor can help them to go to the site of actual effect, and this phenomena is actually what you call homing. So homing is very critical phenomena to direct the activated T cell to the site of injury, to the site of action, or to the site of actual effect. So here is an example. If we understand this, then it's easy to understand this example. So vitamin A, which is retinoic acid. Now this retinoic acid can cause this dendritic cell to help and express certain kind of receptor. Now this retinoic acid is actually a kind of a cytokine which can go and activate the nearby cell also. So basically this is some sort of an activator. So antigen presenting cell is actually binding to this receptor here, and this is again activated by a T cell. Now earlier the T cell was naive, now it is activated. Once it is activated, it will go and do an effect. So this particular phenomena is very critically a homing expression in the gut. So in the human gut, we see that expression of these factor got increased, and this too, say alpha 4, beta 7 and CCR, chemokine receptor 9, can help this to this activated T cell to go towards the gut area to do the effect. That means vitamin A could regulate the homing phenomena by upregulating the gene expression, and these gene expression can do an expression of these specific 2 proteins which are listed here, and this is just an example. There could be different other types of protein which could be responsible for homing towards the lung, towards the cell tissue types. So this is what is a gut homing. So one can easily see that alpha 4, beta 7 and CCR9 is gut homing specific receptors. So this is all about homing. Let's try to understand another cellular phenomena which is called inflammation. Now inflammation is a very general term. People always use this term in order to showcase any kind of an effect that human body does in order to fight with an external stimuli or any kind of injury, toxin or anything that is happening in the human body. Human body does respond to that external stimuli, and all those responses are collectively called as inflammation. And these inflammation are again very important cellular processes in order to support the cellular homeostasis. There are 2 types of inflammation. One is called acute, which is very instantaneous, sudden, and the other is called chronic, very late. Say it could be a days and months and sometimes years also. So acute infection is sudden, very short term response to injury or infection lasting for only few days, and this could be an example such as a cut in the skin area or a sore throat. While chronic is chronic name itself indicates a long term. For example, infection of Mycobacterial tuberculosis can do a long-term inflammation in the lung or any other areas where the bacteria can replicate. It is very slow process, therefore it is long-lasting, and there is a persistent inflammation. So the cells become persistently inflamed because of the long-lasting injury to the site, because the body is unable to remove that causative agent, and therefore the body response is continuously happening, and therefore the chronic is a long lasting thing. So it lasts for the months and years. I just gave an example of the infection of Mycobacterial tuberculosis, TB. Coming to how this inflammation happens in the body and what are the cardinal signs of inflammation. So we need to understand the method or the steps how this inflammation happens in the human body, and what are the cardinal signs of inflammation. When we say cardinal signs of inflammation, that means there are set forth of rules which a body has to follow in order to showcase that there is an inflammation. If anything which is absent from that all cardinal signs, we can't call it inflammation. So let's try to understand the steps of inflammation. So the first step of inflammation is recognition. So I'll just draw here and try to show what this inflammation. So suppose this is a cellular surface or a skin surface, outer surface, and if there is a break here in the cell surface, for example a injury or a cut or a abrasion or anything, this could lead to the rupturing of the cellular area here. And if there is a rupturing of the cellular area, this could damage, or this could lead to the production of many chemicals, and this is the second step, which is release of mediators. And because of this stuff, the chemicals are getting released and going downstream or percolating into the lower stream. Now remember, here we have tissue space, and then here we have a blood vessels where the blood is flowing in, say for this, for just understanding, this is the forward direction of blood flow. So the first is tissue injury, and because of tissue injury we can see that lot of neighboring say resident macrophages cell or any other cell type is also attracted towards the site. So say macrophages which are having a high capturing capacity of antigen can go to the site. So these are resident macrophages which are present already in this tissue space, and because of this injury, then these chemicals are secreted out. So this is a chemical mediators, and some of the examples are histamine, prostaglandins and other cytokines. The third phase is vesicular phase. So vascular phase is actually the phase where the blood vessels are involved. Now once these chemicals are touching to the blood vessel, this can increase the permeability of the blood vessel. So the blood endothelial lining is getting breached, and the fluid is coming out into this area, right. So there are a lot of fluid which are oozing out from this area. Along with that, the phenomena which we have discussed, the rolling of leukocyte diapedesis, so the blood cells can also come out into this tissue space in order to fight with the infection. So there could be a leakage, and there could be a migration of cell towards the site of injury, and these can go here to start the effect. So this is a vascular phase, and I already explained the cellular phase as well. So white blood cell, neutrophils and macrophages move from blood vessel to the site of injury to destroy the pathogen and remove the dead cells. So essentially these two phenomena can cause say release of the cellular content as well as the liquid content of the blood vessel to the site of injury. And finally we will see that because of these all processes altogether, repair will start slowly. So this is the overall phenomena that's happening. So I'll just summarize it one more time, and then we'll understand it in detail. So I'll summarize it one more time. So the injury could lead to the breach in the cellular system or in the skin area. This can cause the activation of resident macrophages and also the release of cytokines or some chemicals which can slowly percolate down into the blood vessels. This blood vessel, after touching or after getting a signal from these cytokines, will cause increase in vascular permeability. The content will come out from the vascular area to the site of injury, and also the cells will come out. So these two can cause the release of lot of other material which can start the healing process. Now if we see this whole process, what we could observe from outside, that because there is a high level of flux of the fluid, we can see there is a swelling. So the first thing is we will see the swelling, and then we can also see, because the RBC is also involved in this site, the blood can also rush towards the site. We can see the redness is also coming, and when the blood is oozing towards the site, it also carry heat along with it. So one can also see heat coming out into the site. So there could be a swelling, there could be a redness, and then heat also will be quite visible into this area. Now apart from that, the nerve ending which are present in this particular area will be sensitized, because the body system can't be without a nerve system. So the nerves which are having their ending here will get sensitized, and we could see that because of this inflammation we can observe pain. So this pain phenomena is happening because of the sensitization of the nerves around that particular site of injury. And finally, the whole area become functionless, so we can call loss of function. So swelling, redness, heat, pain and loss of function, these are called the cardinal signs of inflammation. So the cardinal signs of inflammation is all 5 combined together. If there is anything which is missing into this list, we can't call that as inflammation. We could call that something else. So the inflammation is that process where we can encounter all these 5 phenomena all together. So we have seen that process of inflammation, the acute and chronic. We can also look into the steps of inflammation, such as we can have a recognition step, the release of mediator, vascular, cellular phase, and finally repair phase. The examples of inflammation, it's always with the with the tis. So such as otitis, mastitis, conjunctivitis. So these all, hepatitis, these are all term which ends with tis and that denotes the inflammation. So any word, particularly in immunology or in the microbiology, where we have tis at the end of the word, we can call it suffix. We can always say that this is an inflammation. And some examples could be hepatitis, conjunctivitis, gastritis, stomatitis, which is the inflammation of mouth cavity, arthritis, which is again the inflammation of joint cavity. So these term of tis can define the inflammation, and the word before tis can define the organs type where the inflammation is happening. And finally, we'll just see one more orchestrated, and finally we'll see that how this tissue injury can happen in neurons. Just an example. So suppose if there is a damage to the neuron, and you can see that here the dendrites and the area of axons are getting some kind of a degeneration, and because of that we get an expression of this damage associated molecular pattern, which we have discussed in the last class, and one such example was high mobility group box 1 protein. So this high mobility group box 1 protein can bind to a specific ligand present or a specific type of sensors present in the cellular surface, and for example here it is toll-like receptor 4. So remember, when there is a damage to the neuron, the HMGB is expressed or released from the damaged cell, and this HMGB can bind to the cellular sensors such as toll-like receptor 4, and it can promote the inflammation. So therefore the inflammation can be started by this process, particularly when we are talking about neurons. Now how a healthy neuron can behave. A healthy neuron can have something called acetylcholine, and acetylcholine is just a neurotransmitter, and neurotransmitter are specific chemicals which are secreted in between the two neurons or synapse, where we see that the neurons can pass on information to the other neurons where some chemicals are secreted. So these are called neurotransmitter, and acetylcholine is one such neurotransmitter. So when that neuron is healthy, the acetylcholines are secreted. This can bind to the acetylcholine receptor present on the cell, and this can inhibit the inflammation. So essentially we could see that this is a negative aspect. So this is a damage associated molecular pattern. So one can always see to avoid the expression of HMGB in order to inhibit the inflammation, and then one has to see that the expression of acetylcholine increase so that to restore the health of a neuron. That means this is a negative aspect or negative molecules which can lead to inflammation, and this is a positive molecule. This can heal the inflammation. Thank you.